about atherosclerosis. So how did I get to signaling? I started off at Alex Levitsky lab doing signal transduction. My focus of my PhD work was to generate inhibitors for IGF-1 receptor, tyrosine kinase. And then I shifted to Stanford and moved to the protease field, where I decided to stay. I love this field. So proteases, how are they involved in signaling? For many years, it's just been thought as degrading enzymes and not really involved much in signaling. But more and more information emerging saying that proteases are now seen as extremely important signaling molecules that are involved in numerous vital processes. And if we could look at the different processes that we already know that they're involved in, cell death, apoptosis, necrosis, I think anything that we just think about, you could see the involvement in proteases. And I'll explain in a few minutes why this happens, but it's obvious that these enzymes could regulate different processes because what they do is cleave other enzymes. And if those are involved in signaling, then we have some kind of regulation. So post-translational modification, cell death, proliferation, immune response, DNA replication, and it goes on and on. So this is a school. Not that many students here, but still. I'll say a little bit about proteases. So proteases are generated in the cells as inactive zymogens, and that is because what their outcome or their activity could be a degrading, or it could be very harmful to the cells. So they're generated as inactive zymogens, and then they undergo activation processes to generate an active enzyme. And those processes could be cleavage of a little inhibitory part, or dimerization, or shift to a different spot in the cell. And at the end, we have an active protease. And this protease could now cleave its substrates. How does it know what to cleave? It's upon the sequence of that protein. So that protein has a specific sequence, and the protease can recognize that sequence and cleave the amide bond, and then generate fragments or cleavage products. Now, a single protease would recognize several sequences. It's not just a single sequence. So it could cleave many substrates. And there are some of these proteases that that cleave that many different proteins, like caspase 3 could cleave so many substrates in the cells. And there are other types of proteases that are very selective to specific ones, okay? But since this could be so detrimental, there are also endogenous inhibitors could now block this activity and regulate the activity of these enzymes. Okay, so different proteases are called or termed by the amino acid or the residue that is actually at the activation. So we have serine proteases that have a serine at the active site, cysteine proteases, aspartic proteases, and metalloproteases, which actually have a metal, but the actual driving force, or actually the nucleophile, is a water molecule. And the idea behind these molecules are trying to figure out where they're active, because they could be inactive and they could be inhibited. And what we're really interested are the activities of these enzymes. So in a few slides, I'll show you our approach to learn about these enzymes. But first, I want to show you a little bit about a family of these. Oh, and I forgot to say that the reason that these enzymes are involved in that many processes is because about 1 to 2% of the human genome codes for proteases. There are really that many of them, about more than 550 proteases in the body. So they're really involved in so many things. And I think that if across the board, if you think about any of the proteases you know, you would know at least a few of them, because they're involved really and a lot in cancer, in the immune response, and whatever you guys do. So one family of proteases that I want to talk about are the cysteine cathepsins and their involvement in cancer. The cysteine cathepsins are a family of 11 proteases. They usually reside in the lysosome of the cells. However, in various pathological conditions, they could be secreted out. For instance, in cancer, they could be secreted out of the cell, and then they contribute to migration of the cancer cells, metastasis, and proliferation. If we look at the cathepsins here in this model of a tumor, you could see that they're involved in activation of different proteases. These are the matrix metalloproteinases. They could be involved in degrading the extracellular matrix, and they could also be involved in cleaving, like cadherin, and releasing cells and inducing migration. If we're looking about about looking on these enzymes and where or which of the cell types express them heavily, then we know that the macrophages, or more specifically the tumor-associated macrophages, highly overexpress these enzymes, both internally and secreted out. Okay, so these family of enzymes are highly important, as others, but I'd like to focus on these. And I want to show you a bit of the tools that we generate to learn or to study or image the protease activity. So the idea is to take some kind of recognition element, which would be a sequence that the enzyme recognizes. We'd attach a tag to that, and we could choose any tag that we want to slap on there. Oftentimes we use fluorescent tags, but I'll show you by the end of this talk that we start to use different types of tags like contrast agents for CT, and that's what I'm going to be showing you, but it could be a radioactive tag. So as chemists, and I am a chemist, we could choose which one of these tags we could put on, because these are usually small molecules. And then what we have is very important in these types of probes is a warhead. That's the term in the field. I didn't term this, but the warhead is some kind of chemical moiety that enables a irreversible covalent linkage with the enzyme. So what we have here, we have an electrophile that now when the nucleophile of the protease is going to try and come and cleave this substrate, because it thinks it's a substrate, it will latch on to this probe, and now we'll label it with the tag. And then we could see where that enzyme was active. Okay, so this is how it looks down to the molecular level. This is a substrate, that's the tag. Here the enzyme, we're talking about cysteine proteases, so a cysteine in the active site. Cysteine thinks it wants to cleave it, but now we have this acyloxymethyl ketone. So this AOMK is right there. It binds to it. It forms a thioether bond that's now stable, can't be hydrolyzed, and I have a covalent linkage between the tag and the enzyme. And now we could tag where these enzymes were. So these types of probes were there when I started my postdoc.
Doesn't it interfere with the activity of the enzyme?
It does. It inhibits the enzyme, and now the enzyme's dead. But I know where the enzyme used to be active, so I could study it. Absolutely. This is a suicide inhibitor. Okay, so when these types of probes were there when I started my research, and then in Matt Bojo's lab together, we thought about this cool idea of generating a sophisticated probe. And the reason was because when we have a probe that has a fluorescent tag on it, it's fluorescing all the time, bound or not bound to the enzyme. It's fluorescing. And in order to see where it's really bound to the enzyme, we need some kind of purification step or some kind of wash or whatever to get rid of this free probe and only see the tag probe. So we came up with this type of probe, which is called the Quenched fluorescent activity-based probe. And this is my postdoc research. So it's a tag, it has a linker, it has the same AOMK, and we put a quencher on at the end. And now, when the enzyme comes in, it binds into the binding pocket, the protease tries to cleave it, it attaches to that AOMK, it releases that little piece off, and that piece now has a quencher on it, and now this starts to fluoresce. And now we could see the switch. We could see where the enzyme are active right away when this actually happens, okay? So it's real time. And we did this, first we showed this in vitro, and then we showed this could work in vivo, we could look and see these active macrophages within a tumor, and we could actually spot them using non-invasive imaging, or this was fluorescent imaging. Okay, so that's what we published here. And this is one of the slides I just like to show that will give you a little a touch of how this works. So we have these animals that are tumor-bearing mice. They have MDA-MB231 breast cancers on their back. And these are all expressing green, so they have GFP. We inject one of these red fluorescent activity-based probes into the animal. And when we resect our tumors out, we could see that there's a lot of red, which is our probe, a lot of this red probe here, and it doesn't really actually co-localize right onto the tumor cells, because what it's actually seeing when we FACS sort it, that these are the tumor-associated macrophages, and they highly expressed. If we do look careful, we see a little bit of the proteases in tumor cells, and many papers say that tumor cells have higher cathepsin activity than normal, and they do, but it's nothing compared to how much the tumor-associated macrophages have. They have a lot, and it's in the cells, and these are small molecules that actually get into the cells and label them fluorescent. So we have that. So starting my lab here at the Hebrew U, the thought was, okay, let's learn a little bit about macrophages in atherosclerosis. And the idea behind the project, and I am very towards applicable stuff, so the idea was, let's look at atherosclerosis. So this is a disease of many years in which we have accumulation of fat on tissue that usually happens where the bifurcation is in our arteries. We have a little injury in, or I could say some injury in the endothelial cells. We have some fat deposit inside, come in a lot of these immune cells, and the fat gets oxidized. Fat gets oxidized. We have a plaque coming in with a lot of the immune cells coming in. We have lymphocytes. We have leukocytes. And what happens is these cells actually that respond to the oxidated lipids actually secrete these enzymes that cause rupture of this plaque. And this rupture actually gives a clot into the bloodstream, and this is actually the harmful point of atherosclerosis, when the clot goes away and then it blocks somewhere. And my thought was, if we have a way to determine where we have active macrophages, then we have a way to determine where a plaque is going to rupture. This is specifically important because for many years it was thought that there are two types of plaques, or we could divide them to two types. One is called a stable plaque, which would not rupture and usually is more safe for the patient. And the other one is a vulnerable plaque. And originally they thought that the stenosis, the amount of blood that could go through the artery, is actually determined onto if this is a vulnerable plaque or this is a stable plaque. But it's not that it actually determines the difference. The difference is now known to be the amount of immune cells within the plaque that could actually lead to rupture of the plaque. And we wanted to use this as a tool to go ahead and differentiate between stable plaque that will not rupture or vulnerable plaque that would kill the guy or the girl. Okay, so that was our idea. What we did is we collected tissue from plaques that were resected from the carotid artery. So this is a human patient. You could see the clot right here. What you could actually see here in the white spot is a calcification on the plaque, but you could also see how the blood flow is restricted right here. And so from this restriction, you don't know which type of plaque this is. You only see the stenosis of the blood flow. You don't know if the plaque is going to rupture or not. So what we did is we took out the plaque, and I just want to step aside and say, this is the plaque. I'm holding it. Look at the size of this thing. This is in the carotid artery right here. This thing is enormous. It's the devil. It's full of fat and inflammation right there. It's hard to see from this lighting, but it's really big things that are taken out of this artery right here that leads to the brain. And if it's clotted on both sides, so this is the endarterectomy. This is the actually way of taking this plaque out. So they tie these types of the artery right here. They cut the blood vessel. They scoop it out with a spatula, and this is what I'm holding right there. And then they sew it up. Usually they put in some kind of patch in there to make it a little bit larger so it won't clot again. And this is the surgery that patients undergo oftentimes when they have carotid arteries. So what we did is we collected these arteries, these plaques from the patients, and these are how they look. So you see that they're actually cut because this is how they're removed. And we gave them to a pathologist that segregated them into different types, 1, 2, and 3, depending on the progression towards a vulnerable plaque. Okay, and what we can see here, we're missing all the labels here, but this This is cathepsin. This is CD86, which is saying the immune cells. This is cathepsin B and cathepsin L. And you can see how the plaque progresses from plaque that is stage 1. This is plaque, but we determine stage 2, and plaque stage 3. So when we have stage 3, we have a lot of immune infiltration. We have a lot of cathepsin activity. Sorry for the labeling. I don't know where they went. Okay. So what we did is we used our activity-based probes now on these types of plaques that we could determine as type 1, grade 2, and grade 3. And you could see in how these bands of the different cathepsins, and these are cathepsin B and S, in these plaques, how they go and there's more activity of these enzymes. As I said, these activity-based probes covalently link to their enzymes. So I could run a gel on, this is just an SDS page. I could run the lysates of these tissue on a gel, and since our fluorescent probe binds to our enzyme, we could actually reside the different activities and the different proteins that are bound to these enzymes. And here you can see that there's their activity in the grade 1, but as we go to the grade 3, we see higher activity. And just as controls, we took what we call near plaque. So we took tissue that is not plaque. It's supposed to be as much as we could get to normal tissue, because you can't get normal tissue out of a carotid plaque, right? But so this is near plaque tissue, and this is the tissue itself, and we could actually see the amount of protease activity go and intensify in these plaques. We saw how cathepsin S and cathepsin B activity is much higher in symptomatic patients, and you could see that with the different grades and the different proteases. So we were very excited about this use of our activity-based probes to actually try to determine which of the plaques are for patients are symptomatic. Now, I also want to take aside 1 minute. This inhibitor, I'm going to be talking about it for quite a bit of this talk. This is an inhibitor of these proteases, and it's basically the basic structure of our activity-based probe. So it's the same thing. It's the same suicide inhibitor, but now it's not labeled with a fluorophore, and it's highly potent against these enzymes. As you can see, it really inhibits the activity. If we compare the one without the probe, we see the activity. With the probe, we see with the inhibitor, we see the activity reduced. Okay, so the question is, what do these enzymes do in these plaques? Why are they so elevated? And what is it? What do these enzymes, or what are they involved in? Okay, so this could be a tool to detect vulnerable plaques, but how are they involved in the pathology itself? So this is the part of Tomi's PhD. He's about to finish it. So this is a submitted paper that we're just now hoping for it to be accepted. So we're looking on autophagy. Since we know that these enzymes are in many pathologies secreted, but also they reside in the lysosomes, and they are known to be involved in autophagy, we were thinking to look at how they influence the outcome of autophagy. And I'm not going to go through this because Adi gave a very nice introduction about autophagy, but just so we could follow this talk, if we're looking at the phagophore that now has some of the cell components in it, we form throughout the development of autophagy, there is an autophagosome that has basically differing proteins on the membrane, including LC3 and P62, which actually brings or targets other proteins to the autophagolysosome. This one now, fuses with a lysosome, and we have an autophagolysosome that will actually produce or degrade or recycle these proteins in the cell that need to be recycled. So this is well known in the field, and I just want you guys to think what happened if I would inhibit this process at one point, let's say at this point. What would we expect to see? We'd expect to see more of this LC3 and P63 in these organelles in the cells, because we now halted the outcome or the mature outcome and degradation and recycling of the autophagy process. Okay, so this is just for us, and I think that Adi spoke about the rest of it. Okay, so first of all, we wanted to see what happens when we have primary macrophages that were treated with LDL or regular acetylated LDL. If we look at the cathepsin activity, this is a quenched fluorescent cathepsin activity-based probe. It's dark when there's no cathepsin activity, and when the cathepsin activity is there, it lights up. So this is a micrograph. We could see that there's very little cathepsin or baseline cathepsin activity when there's just control macrophages. And when we treat with acetylated LDL, we see an elevation in the cathepsin activity. And we see that this co-localizes very nicely with LAMP1, which is a lysosomal marker. And we could quantify that. And then we wanted to see what happens when we add oxidized LDL. And we see that there's a significant reduction in the cathepsin activity. So something in the oxidized LDL reduces the cathepsin activity from where the acetylated LDL actually caused it to be. Okay, and this is what we could see here in the quantification. So if we look just at the autophagy with this type of experiment, if we look at the control cells, we see a minimal basal amount of autophagy. And if we add the acetylated LDL, this really causes the cells to amplify their amount of autophagy. This again could be quantified, but this is in cell culture. All the rest is done in primary macrophages. So when we treat them with cholesterol, we see an increase in the autophagic vesicles. So we chose 7-ketocholesterol as a major component of oxidized LDL, and now this would be our model for oxidative LDL, and we treated ourselves with the 7-ketocholesterol. And if this is the basal activity of cathepsin activity, if this is the basal cathepsin activity, we see that it actually is reduced over time. And since we could resolve them on gel, we could see which of the cathepsins is inhibited, and we see that cathepsin B and cathepsin L are inhibited within 2 hours of this 7-keto cholesterol treatment, and even cathepsin S is inhibited, but a little bit slower. So something in the 7-ketocholesterol or somehow downstream is causing the cathepsin to be inhibited. And how this reflects the outcome of autophagy, that I will show you in a minute. But if we look at the cathepsin activity, just the control cells, this is the basal activity. And if we look at the 7-ketocholesterol treated, we see that basal activity is now reduced. So 7-ketocholesterol it inhibits the cathepsin activity. Okay. And this is how we read our autophagy. If we look at the autophagy, we could see, so what we have here are two types of cells, either one that were treated with a vehicle or one that were treated with the GB111 with that cathepsin inhibitor for 16 hours. And then on top of either this or that, we treat with 7-ketocholesterol, and we look at the different components of autophagy, LC3 and P62. And you could see that first of all, just the basal addition of the cathepsin inhibitor would increase the autophagy. And this is, I think, very straightforward. Like if we have the autophagolysosome and now we're blocking the outcome, it cannot degrade the enzymes. We're stuck, and we have an elevated amount of these LC3 and P62. So these are the two that we can see here. And now when we add 7-ketocholesterol with time, we see the increase. Well, at least for 4 hours, we see a nice increase in both of them together. We could also see this very nicely in the P62 microgram. Okay, now we'd like to see if there's actually co-localization between the lysosome and the LC3B, labeling the autophagosomes. And we could see that with the treatment of both the ketocholesterol and the inhibitor, we see an increase in the co-localization between the lysosomes and our now this is LC3B. So now we really see that there's some kind of halting of this process of autophagy. Okay, so now what does this cause? If we're now halting something that the cell would like to replenish now new organelles, how does this, what is the outcome in the cells? So we tried to use a shotgun proteomic approach. We looked at a few of these enzymes. We saw a few, and what we really see are different proteins that are involved in autophagy, like the LC3, like the P62, and obviously other proteases themselves are now elevated because they process each other in this cycle. Okay, and what is the outcome of that? So we went, first of all, this. So we wanted to look at how this influences mitochondria or mitochondria function. It has been known from different publications that when we treat with an oxidized cholesterol, we see impaired mitochondria. So we're trying to see if this actually, the treatment of the cathepsin inhibitor would actually impair mitochondria function or the outcome of mitochondria. And what we could see here is a very nice colocalization between the lysotracker, which tracks the lysosomes, and the mitotracker. You can see that in the control we see separate compartments, but when we treat the 7-ketoglycerol, we see them colocalizing, meaning that the mitochondria is actually targeting to the lysosomes. And now they're supposed to be degraded, but they're not, because we're inhibiting, well, we will be inhibiting the cathepsins, and they're stuck. And then what happens here? So this is the colocalization of the LC3B with the mitochondria, and this we try to quantify this. So although the colocalization percentage is fairly low, we still see an increase in the colocalization of the, autophagosome and the mitochondria when we treat with both the inhibitor of the cathepsins and the oxidized cholesterol. Okay. So how does this really, what happens to the mitochondria when this happens? So we can look at the circularity of mitochondria. So when a mitochondria is intact, it's elongated, and when it's impaired, it's circular. And you can measure by microscopy the circularity of the mitochondria, and you can see an increase in the circularity of the mitochondria when we treat with the cathepsin inhibitor. So we're not letting the mitochondria recycle like it should be, and that would cause ROS. Okay? So we could look at the mitochondria reactive oxygen species, because now the mitochondria is impaired, and you can see that that goes up when we treat with our inhibitor and together with with the inhibitor on its own, but with the inhibitor with the 7-ketoglycerol, this is even more amplified. Okay, so if we go to the seahorse experiments and look at the function of our macrophages or primary macrophages that are either treated with the cholesterol, with the cathepsin inhibitor, or with both, we see impairment in all of the functions that we looked at: the basal respiration, the ATP turnover, and the spare respiratory capacity. So what happens if we have now elevated reactive oxygen species and mitochondria are not working? What do we get? The outcome is promotion of pro-inflammatory phenotype. So we could see all these different cytokines go up as we treat with our, specifically if we treat with the red boxes with both the 7-ketoglycerol and the GB111. Why am I saying this? Because oftentimes when you want to treat atherosclerosis, there was a thought, and there's a lot of literature about this, that now is a good time to treat with the cathepsin inhibitors, because the cathepsins are secreted of the cell and they degrade the extracellular matrix and they actually enable the plaque to rupture. But we have to keep in mind that there's other things that happen here. If we inhibit the cathepsins, we now we could elevate the pro-inflammatory response, and this could be detrimental. So we should be very careful. So I think this is the major outcome of what I'm trying to show you here, that it's not very straightforward. So we did this in the primary macrophages. We also did this in human macrophages, and we saw very similar results. So we see elevation in IL-1 alpha and TNF alpha, MCP, and the rest. Okay. So to conclude this part, dysfunctional cathepsin activity has a key role in nascent plaque development, accumulation of dysfunctional mitochondria that leads to vascular inflammation. Cathepsin inhibition in hyperlipidemia settings perturbs mitochondrial dynamics, increased mitochondrial damage, and consequently elicits an inflammatory response. And our data suggests that cathepsins are paramount, important for cellular quality control mechanism, and highlights their importance in cases of cardiometabolic disorders such as hyperlipidemia and atherosclerosis. Okay, so that was my part of signaling. And now I can show you some of the applicable stuff. So these are basically the tools that we could play around in the lab. So we have our different types of probes. What we do put on our tags is optical tags, as I think I just showed you some of that. CT contrast agents, which I'll show you at the end of this talk. And we apply them to different diseases now that we're still working out. And I want to show you a little bit more of some new tools that we generate. So as I said, I have a background in chemistry, and one of my thoughts were, when I just started my lab at the Hebrew U, was if we could get these probes to the macrophages of a tumor, and we could label them, and we could see them, why can't we use those to actually target those macrophages and try to deplete them or ablate them? So we came up with this probe. So this probe is a photosensitized quenched activity-based probe. So instead of using just a fluorophore to detect where these proteases were active, we would now use a specific protease, which is called a photosensitizer. So it's, a specific fluorophore, which is a photosensitizer that on the one hand gives fluorescent light, and we could detect it, but on the other hand, when we shine light at it at a specific wavelength, which it absorbs, it will generate reactive oxygen species, which would actually kill the cell. So we could use this as some kind of switch to target these photosensitizers to the cells, to the tumor cells, or the macrophages, or tumor-associated macrophages. But on the other hand, the reason that people do not use or don't use this in all types of cancers is because these photosensitizers are very harmful. So if I give someone a photosensitizer, the skin gets now light sensitive. And if it would go out into sunlight, we'd get burns. But if we generate a quenched photosensitizer activity-based probe, so this photosensitizer would not generate reactive oxygen species because it's quenched by a quencher. So it would give the energy to a quencher instead of giving the energy to an oxygen molecule generating signaling oxygen. So this one would be safe, while this one, after bound to a protease releasing the quencher, now could kill the cells. So we have a few, I think, nice advantages of these types of tools. Sorry for Magave life. Okay, so I just want to show you the types of experiments that we could do in the lab. So these animals have 2 tumor spots on the back of the mouse. We can inject these types of quenched fluorescent photosensitizer activity-based probes to the tail vein. These compounds will circulate in the body of the mouse. They will bind covalently to the enzymes in the tumor-associated macrophages. You could see that these are quenched. So 10 minutes after injection of this type of probe, it's fluorescent probe, but it's quenched, so we don't see the fluorescence. It will light up wherever we see proteases. So you see some here. This is probably either the spleen or one of the kidneys that do have cathepsin activity in them. But it will also light up the 2 tumors that we have. Then we had one of the tumors exposed to light. Okay, so this one is the left tumor on this side. And the other one was kept in the dark in the most sophisticated way. We could keep one tumor in the dark and the other we could shine light. We just covered one tumor with tinfoil. And then we let the compound keep on circulating. After 16 hours, we could have another light treatment. And at the end of the experiment, we could resect these tumors and see what we get. And as controlled, we use a bacteriochlorine, which was a photosensitizer. It's sensitive to high wavelengths, so very low energy, 760 nanometer light. And when we treat these, you could see that by the time we did the 8-hour treatment, this probe that's fluorescent is way gone out of the body, and so we wouldn't expect much of this photosensitizing to happen. So this is the outcome of the tumors. We could see the tumors that were treated with vehicle they're not harmed, what we could see here is antibody for cleaved caspase 3 as an apoptosis marking. We see the F480 as a macrophage in the merge. We don't see any apoptosis here. We see a lot of macrophages here. If we just use the bacterial chlorine by the 24-hour mark, there was none there left, so no death. But when we use our activity-based probes here that now covalently link this photosensitizer to the tumors, only in the light treatment we see massive apoptosis, and we could see that this apoptosis is co-localized very nicely to the tumor-associated macrophages. So this is how the outcome is just looks. These are animals that were treated with the light and the photosensitizer or activity-based probe. You can see that either the photosensitizer on its own or without light, or just light without the photosensitizer activity-based probe doesn't do anything, but to the tumors you could see impaired growth. And yes, there is still some growth, but it's a single injection of the compound just followed by 3 light treatments, so this could be improved. Okay, I want to show you another tool. Wow, it's really hard to see in this lighting. So this is an activity-based probe that's targeted to the nucleus. You could see this go into the nucleus. So there's a lot of publications in the literature showing that, yes, I'd like if you could close the light because there's another one of those. I could try showing this one again to you. You could try. But there's quite a few, just for 1 second, and I'll ask you to keep it there. Keep it there. So there's a lot of literature about cathepsins active in the nucleus. So we were thinking we could generate a nucleus target quenched activity-based probe. When these proteases would shift to the nucleus and try to cleave their substrates there, it would then light up. So this is our attempt. We generated a probe that actually has a nuclear localization signal to it. And now you can see that the red are the activity-based probes, and you see that over time, this is a time-lapse movie over 10 hours. You could see that these enzymes actually go and reside in the nucleus. And that was great because we could actually see what was happening. But then I was thinking, if you attach an NLS to a protein, it will bring it to the nucleus. So who said that that's a protein going to the nucleus on itself, or am I dragging the protein to the nucleus? So we did the same experiment with a compound that doesn't have an NLS, and this is what you could see. So it's not as bright because it doesn't get as much into the cells. You see the activity-based probe around the nucleus right here. If we focus on this cell right here, you see that it actually goes into the nucleus a little bit, and then the cells I thought was dying. No, it's not dying. It's dividing. This is mitosis. You can see this with the other cell up here. They're also mitosing. This one also is undergoing mitosis, and so on and so forth. And this is a project that's now under research in the lab. So we're still using these types of tools to investigate that. There's one more microscopic movie, and then I'd like the lights back up before everybody falls asleep here. This is a quenched activity-based probe used for caspases, because caspases are also cysteine proteases, as their name says. So this is Yulia's project. We tried to look at caspases. These are cells that were treated with cisplatin. We wanted to see what happened to the caspases in the cells. Where do they cleave? Where are they active? You could see that we see these spots in the cells, and they're not just diffuse, where they're probably also diffuse. We could very well see the specific spots in the cells where these caspases are active, and then the cell undergoes apoptosis. And when we actually investigated a bit more on that, we saw that the activity of caspase 3 is in the ER during apoptosis, and this was a new finding that we could only see using these types of tools. Mickey, thank you. If you could turn on the lights, thanks. Okay, so yes. Okay, so now I'm going to show you our recent stuff that just got published in JACS on our CT activity-based probes. So the idea of CT is widely used in the clinic, as we all know. It's usually used for anatomical imaging, but and it also gives very high resolution images. But for contrast, there is a problem because it has very, very low sensitivity. This is a project of Galia and Hammond in the lab. And so we decided that we have this massive accumulation of these compounds. We have these massive amount of active enzymes, tumor-associated macrophages. Why don't we generate these probes that are labeled with a CT contrast agent? Now they will bring these contrast agents to the tumor, and we could say something about the tumor progression and where the tumors are, and hopefully one day we might see MITs. We're working on that. So first thing was to see the sensitivity of the CT machine to the contrast agents, because we know it's not sensitive to contrast. So we use gold nanoparticles, 100 nanometer size, gadolinium and iodine. And when we see that the gold, while it's a little less sensitive to gold versus iodine, we see the attenuation of gold rising very quickly, so we don't need that much that we could see. On the other hand, we see iodine, so we have these two projects. One is on gold, one is on iodine. I'm just going to present the gold because it's getting late. So these were our structures. These are gold nanoparticles, and we didn't really know how we're going to link them up. What is the coding? What is the best ratio between targeting moiety here represented in red versus a gold nanoparticle? Should we put 10% targeting moiety? Should we put 1 targeting moiety per gold particle? This is a big particle. Should we put 50? Should we put 100% of the targeting moiety? Which one would be best? Maybe this one will attach 3 enzymes and we'll lose our or we'll lose some of the signal. And then we read a few papers saying that if we put the targeting moiety in a shorter peg, we get this mushroom configuration that actually gives more activity. So we played around with the different coatings, and we played around with the sizes of our gold particles, and we generated a few of these compounds, and we went out to test them. So what we saw is, first of all the particles, if we now just quantify this per amount of red spots, which is the GB111, which is the same inhibitor, which is actually the concentration of the inhibitor per each gold particle, we see that most of them actually have the same activity. How do I see the activity? We treat this enzyme, okay? This is just a cathepsin B enzyme. We treat the cathepsin B enzyme with our probe as an inhibitor, and then we come in with a fluorescent probe looking at the residual activity, and we could see that if that residual activity goes away, that means that our probe actually bound to our target. So we could see that they all do the same. This is a control that doesn't have any targeting moiety, so we see that nice. So you see that we're all more the same, but the question is, could these big gold particles actually get into the cell? I mean, we're talking about 10, 30, or 100 nanometer particles. Do they get into the cells? And the answer is yes, they do, to my surprise. But they get into the cells depending on the size of the particle. So if we look at the 10 nanometer particles, we see that they all more or less get into the cells. The best one that we found later to be is version 4, which is version 4 is actually the best, but you could also see this here. See, well, do we have time for this? How much time do we have? 5 to 10. So I won't go into the story, but they accumulate in the lysosomes. They inhibit within the cells. They inhibit more than they inhibit in vitro because they just gather in there. They accumulate in there, and you see that the smaller particles inhibit better than the 30 nanometers, and the 100 nanometers are the worst. And we wanted to know how this would work in vivo. So in vivo, we have tumor-bearing mice. We put a tumor on the back of the mouse. We injected with our version 4 of the compound. And then we followed over time. We didn't know what the kinetics would be. So we followed over time before injection and every day after injection for 3 days. Then we took, these are CT images. Then we looked at flame atomic absorption spectroscopy that would just tell us how much gold we actually have in the tissue. And then we took all these to electron microscopy because we could actually see where the gold particles are within the tumor. So this is a long a big amount of work, but what we could see is that so we always have the non-targeted compounds to see just the EPR, enhanced permeability and retention effect, just to see if the basal accumulation. So non-targeted of the 10 nanometers don't accumulate very well. You could see the nice accumulation on the tumor right there. You could see the 30 nanometers and the 100 nanometers don't accumulate as well. So this is, it's a combination of cell permeability and the retention and the permeability into the tumor. You could see that the white circle here is actually the gold. And if you could look down here and you could say, well, there's some in the digestive system. So this is a pre-scan of one of the animals. So you could see these particles in the system, and that's probably some metals that they have in their food. Okay, this is the quantification. So if we look at the different sizes of the particles over time, we could see that the 10 nanometers actually do accumulate most. 30 nanometers as well, 100 nanometers weren't very good. Okay, if we take out the tumors and look at the amount of gold, we see a very similar picture to what we see here. So the 10 nanometers accumulate most, 30 nanometers and 100 nanometers. Now, I want to draw your attention to the y-axis here. What we're actually quantifying this, since the tumor is growing over the 3 days, we had to quantify this in a non-biased way. So we couldn't just take the size of the tumor at the end, because that wouldn't reflect the thing 3 days or 4 days before that. So what we did is we looked at the amount of pixels that we could find in the tissue, and we looked at the percent of the pixels that were labeled with gold. So if you look carefully, over 10% of the gold tissue was actually labeled, 10% of the tumor tissue was actually labeled with gold. So this is a large amount of gold in these tumors. Okay, these are the comparisons to the non-targeted. You see that the targeted really accumulates much nicer than the non-targeted, and when you look at the 30 nanometers, this is highly significant throughout. 100 nanometers weren't as well, didn't work as very well. Okay, I think this is the coolest picture. Really, in my whole talk, this is the coolest picture. Because when you take these tumors and you look at the TEM image that was done by Yael that's sitting right there. So we took these tumors out from the animals that were injected with a probe, and we subjected them to TEM analysis. This is a single cell, okay? This is nucleus of the cell. These are the lysosomes. You know that the lysosomes, because they have double membranes, so they're a bit darker. So you see these are the lysosomes, and if you look carefully, you actually see the particles within the lysosome within the cells, and you see that much more in the targeted particles than in the non-targeted particles. If you look at the 30 nanometers, this is even more pronounced. You see the particles really nicely. They are 30 nanometers in size, way less in the non-targeted compounds. So the last control was asked by the reviewers. They said that this has nothing to do with the activity of the enzyme because there's a different surface area to the particles that have our probe. This is a small molecule. It must have been reviewer number. And yeah, reviewer number 3. So anyhow, we generated, we compared we have now 2 negative controls. One that we just put a peptide that has nothing to do with the cathepsin activity and doesn't have this warhead to it. And one that was pre-treated, well, the compounds that were pre-treated with an inhibitor before treating with the probe itself. So we're supposed to block the cathepsin activity. And to our delight, we saw exactly what we were expected. With a probe, we saw nice accumulation. And with all the controls, we saw reduction of the accumulation. Take-home message is activity-based probe imaging is a platform that can be used as various imaging reagents for various diseases. I showed 2. We're working on a few others in the lab now, but it's really out there for people to use. These are nice tools that you can use, fluorescent or even in vivo. For the CT probes, we have a CT now in the lab, so yay. Activity-based probes could potentially be translated to the clinic because they could be visualized by CT. And we aim to collaborate with investigators that can make use of our probes for new applications, promoting existing applications. This is my lab. This is Daria that showed the project on the gold particles. This is Hanmat that had the iodine. I didn't show any of that. And where's Tommy with the glasses? This is Tommy. It was the project of Tommy on the autophagy, Emmanuel, the technician, Karin that had the nuclear localization, Yael that had the photosensitizers, and to the fundings, and to my world collaborators, and thank you for your attention and for bearing me till the last minute.